Hello. Hi, Sanjay.
Yeah, hello.
Hello, Mainak.
Hi. Yeah, hi.
So, yeah, Sanjay, I talked to you in Slack, so you want to give an introduction to yourself?
Yeah, sure. Just a second. Yeah, I'm good.
Yes.
Yeah, okay. So I'm basically a junior at Amrita School of Engineering, Chennai campus. So I'm interested in deep learning projects for a while, but I'm not to an advanced level. I'm just working on some intermediate level projects right now. So I got to know about the DevoWorm community recently. So I just thought that it could be nice to contribute to the community as well as learn something from the members of the community as well. So I started exploring the projects right now. So I'll start working in a while.
All right, yeah, that's great. Thank you for the introduction. I hope you find something you want to do. Like I said, I think I sent you a link to the onboarding guide, which is what we use to sort of orient people to the group. We have different projects. A lot of times they're like Google Summer Code projects. Other times we have longer term projects that we have. So if there's not something you don't find on the onboarding guide, I can actually bring those up in the channel to make people aware of them because there's a lot of stuff going on, and we'll maybe go through the board later to show what those are. So welcome. Hello, Ujjwal.
Yeah, thank you.
You're welcome.
Hello, hi there.
Hi. All right. And how are you doing?
Pretty fine actually. So now my internship is over, and I'm glad to tell you all like I have got a replacement offer from Deutsche Telekom.
Good.
So now I think I can focus on the projects going on here.
Well, that's good. Glad to hear that everything is going well for you. Your internship sounds like it was pretty successful. And so, we'll be, I guess we're right kind of in the throes of summer of code. So we're getting, to the part where we're really kind of getting some tangible results. So look forward to you providing your input and joining in the group meetings and that.
Yes, sure. Also, Mainak, can you please just copy paste the link once of your whenever you're writing your blog, so I can just get on board what is being done.
Okay.
Yeah, sure. I'll do it right now.
Okay, thank you.
All right, thanks. So yeah, that brings us to Mainak, and he's so that's his blog post, or his blog that he posted in the chat. So that's his Google Summer Code update. He's doing a weekly update at his blog. So Mainak, I checked out your GitHub pushes. They look good. Why don't you give a presentation on your progress for this week?
Yeah, sure. Sure. I'll just go ahead and share the screen and.
Yeah.
Okay. Yeah, can you see the screen now?
It's still blank. It's still blank.
Not yet.
Not yet.
Okay. Okay, I'll just reshare it.
Okay.
Okay, is it visible now?
No.
Okay, so let's retry.
All right.
Okay, so what about now? Is it
It's
visible
still
now?
blank. I don't know.
But I see something like Mainak is blocked, something like that when you.
Yeah, that's what I'm trying to share. Okay, we'll try it once more.
All right.
We'll share the entire screen.
All right.
Okay, so what about now?
No. Why don't you go out and come back in and see what happens?
Yeah, okay, I'll try that. I'll try that in a different network then. Okay.
Yeah. Okay, so he's gonna try to figure out what's wrong with his connection. I hope it's not an issue today, but Well, that's the problem with doing virtual meetings. So
Yeah, like it's pretty difficult. So like this time my internship is also like in completely virtual manner. So it was pretty different experience for everyone. And like I haven't met my mentor once throughout my internship.
okay. Yeah, it's been challenging, but we run this group mostly virtually, so it's kind of Mainak.
Yeah,
Okay. Yeah, you're back. Yeah,
yeah,
I
hello,
can hear you.
okay, sure. I'll just go ahead and share the screen once more. Let's see. Okay, is it visible now?
No, I don't know why it's.
Okay. I guess this is a problem with Jitsi, I guess, because my network shows.
Let
Anyways,
me try once.
Okay.
Should let me try once if I'm able to share it or not.
Yeah. So maybe you can share week six blog post and then I can explain over it, right?
Can you see my screen?
I can't see it.
No, we cannot see the screen.
Yeah, so it's basically some problem with Gypsy, I guess.
Well,
Yeah, this could be a problem with Jitsi, I guess.
all right. So why don't I go through? Why don't I like open it up? I'm doing a recording here. Why don't I open it up on my end?
Yeah.
And if you can't see the share, then you can go back to the video and see the video
Yeah.
or the. Yeah.
I'll see. I can just talk over the points and you can just open it up in your screen.
Yeah. Let me open it up.
It will fit into your screen. The entire blog post will fit in your screen, so the sharing isn't too wide.
Okay. Let me share my screen then.
Okay, so yeah.
Let me try window actually. All right. Can you see my screen
Your
now?
screen is visible. Yeah.
Okay, good.
We can see your screen. Yes.
Okay, so let's go through.
Okay. Yeah. So this is the week 6 update of Google Summer of Code. So the first update, as you can read, the first update was to basically swap out the cell membrane segmentation model that was which was already there in the DevoLearn repository, and I actually had trained a new model, so I had to swap it out and just exchange the PTH file. That's all there was to it. But then I had to run the tests locally because Travis has been they've actually stopped their service. So I had to run the tests locally, which took some time, but it was okay. And in fact, we also have plans to move to GitHub Actions in the next week. So I'll be working on that soon.
Okay.
The image, as you can see, it basically shows the tests which are there. And I'm actually using the PyTest library for that, which again was one of the changes that we had worked on back in the day. So the second point is that I actually had to rename the embryo segmenter package to the cell membrane segmenter because first of all, the name is a bit more specific now because the name itself talks about it. It talks about the function of the package, and the second reason is that to avoid naming conflicts when we add new segmentation models to the library. So on the end of the user, there are only on the end of the user, there is only a very small change which is there, which is the old syntax and the new syntax, as you can see. Previously, the user would have to type from DevoLearn import embryo segmenter, but now the user would just have to change the final word, which is cell membrane segmenter. You would just have to swap out this word, and everything else will work exactly the same. So that was the second change that I made to accommodate for the new segmentation, to actually accommodate for the new model that I would be adding later. So, and then was I actually added the nucleus segmentation model into the library, which I was working on for the past couple weeks. It was trained and it was tested and everything was done. All that was remaining was to add it into the core library. So I actually had to write the functions which were required to run inference on CPU as well as GPU, to make it work with the library as a whole. I wrote all that, and then I made some additions to the testing script too to accommodate for this additional model. And I've actually elaborated the steps which would be required to run inference on one single input image here, and later I'll be working on a function which could basically run inference on an entire file of an entire folder of images, which have like an entire folder that contain input images. So that's something that's coming. And Bradley, can you scroll down a bit? There
Yeah.
are some inference examples.
Okay, here we go.
Yeah. So I was talking about the new nucleus segmentation model. So the examples that you can see on the screen, they're basically taken. These outputs are basically taken from that model itself. So the first GIF, as you can see, the input image, which is basically a 2D image, which is varying through the z-axis. So the inputs are varying through the z-axis, and the segmentation maps are also like the segmentation maps that you see to the right. They're actually corresponding to the input image, which came out of the model, which is basically the model output. So this is one way this is one way the model could be used, or the other way could be that the inputs could actually vary through time too. Like it's not just that the inputs will have to vary through z-axis only. The inputs could be of any form. They could vary in z-axis or in t-axis. So the image below, the GIF below that you see, the input is actually varying through time, and the segmentation maps are also like you can see the corresponding segmentation map to the right. So these are some like I guess these are just examples that one can see to basically understand what's going on and what the model can perform. So can you scroll down a bit?
Yeah.
Okay. So coming to the planned things that I have for the upcoming weeks. So the plans are to actually build a GUI for actually all the DevoLearn models, and to have them hosted online somewhere, to like as an example of, or let's say as a demo, where people could like they could just open up the website and have some examples right there to see basically if it works with their data or whatever. They could have a live demo online without any sort of installation or coding, whatever. So and the next part is to look into ways of running bulk inference from folders or zip files using the web GUI. That would be interesting, because if we can get somehow, like if we can run the bulk inferences on the web app itself, I guess that would be a better thing for a lot of the non-coders out there.
Yeah.
So then the next plan is to move from Travis CI to GitHub Actions, because running the tests offline for every PR or every single little change that I make, it's actually a bit tedious, because it takes nearly 300 seconds or so to run the tests, and like it actually takes time if it's not automated. Like I'll have to go to the terminal and run all that. I have to run the PyText syntax and all that. So it's actually better to have something online that will do the checks for you. So we'll move to GitHub Actions soon. So that's it for the plans actually. And yeah, that's it for this week's update. Thank you.
Oh, good. Thank you. Are there any questions about what was presented? I have a few, but I'll let the rest of the group.
So, like, have you like selected out the platform which you are aiming to like host it and build it, the web app that we are talking about?
Yeah, actually, I have selected. I was running some trial and error, so I basically settled for Heroku only, because we already have a test web app online, which actually runs online. So, because I have the experience of building that test web app, so I think it will be pretty much doable for me to have the other packages of the models to as a web app. So yeah, to answer your question, the answer would be Heroku only. Yeah.
Okay, and like when you say you want to do some bulk inferences, so like do you mean like a person can go and upload a zip file or like something like that to your portal, and like then you will kind of show all the results, all the images combined?
No, actually what I meant is that, like as you said in the first line, that somebody would be able to upload a folder or a zip. Yes, that's correct. Like that's what I aim for. But the output would also be in the same format. Like if the person uploads a folder, he'll get a folder back as the output, which will contain the outputs. So he could, like, after uploading the folder, he'll have to wait a bit, and then after the inference has been run, then I guess he'll have to, after he sees the output folder, he'll have to just download the folder. That's all. That's what he will have to.
So like it's kind of like you are generating all the results and showing some sort of, like, let's say for the sake of argument, like CSV, right?
Yeah.
Whatever the inference is like. So you are basically letting the user download CSV.
Yes. Or an image, if it's a segmentation model, the outputs would be images, so the user would download the image.
Or the segmented images, right?
Yes.
So like how many weeks do you have left now? Like, remaining?
Not much actually. I'm in week 6, so there's not really much time left. So I'll try that. In fact, I guess Bradley could open up the web app, right? So I guess Ujjwal Bhaiya at his cloud that I show the web app, right?
Which web app would that be?
Okay. Anyways, just drop a link to it.
Okay. Yeah.
I'll just drop a link to it in the chat.
All right.
So people can check out the web app.
Because I was thinking like
actually it takes a bit.
I know, but I was thinking something like that in the Hero app. I don't think so it would take much effort, and you can also do it after GSoC as well. So instead of downloading the CSV file that Alice has already done years before, so basically we can show everything on charts in the web app itself. It becomes more intelligent, and like if the user thinks he can download it, obviously the user results, one has to download all the CSV file, and CSV is the most convenient format to use any result. But like before doing that, you can take some sort of averaging and all those things, and you can show statistics on the web app itself. Just like to,
so you're saying to add some sort of plots to have a better idea of the
some sort of yes, some sort of plots on like what is your like, for example, let's say if you are plotting some, you can use Plotly, and like they can be interactive plot where you can show each image has how many cells and like something like that. So it would like give user an overall view before downloading it.
Exactly. Yeah, it's better to see the outputs in a visual format before downloading and checking it. That's a good suggestion. Yeah. So can you see the screen? I was talking about the test web app. Yeah. So Bradley, maybe you can click on any of the input images to the left, and then you could click submit, and then you'll get the results.
Okay, so I'm clicking on this image, submit, and then it's processing, and then it gives you the segmentation and centroid maps.
Yeah. So at this point, he could just right click on the image and download the image. But I guess for, let's say, 100 images, and so for 100 or 1,000 images, it would be sort of it would be very, very tedious to drag and drop every image. So I guess having a folder format would be easier here.
Yeah.
Yes. For the zip format, something like that, I think you can show one or two samples, and then you can just say download. But you are getting a point, right? If there should be some sort of analytics, like a user can scroll down and see, because like I don't think so whoever is uploading the images is much interested in the segmentation images. He will be interested in all those counting points.
The data, yes.
Yes. So if you can show that on this data as well, so it will be a very useful domain.
Yeah, sure. I'll try for that. Thank you.
Yeah, like we are asking, but I don't think so it's going to be a much, work in it. Like you can simply use Properly and it will be an easy task. I don't think it's very tough task.
Yeah.
Once you have parameters.
Yeah, I agree with that. I think well, one thing would be nice would be to have a way to download CSV files. So like to have a numeric something you can download and analyze further. I mean, we can do this technically in like something like ImageJ, but I don't know if that would be better. But maybe just to have like a rudimentary like CSV download, and then to have maybe like report the statistics of the analysis, because if people put in like the perfect image, it'll give you this nice result. But there may be a lot of images, and maybe people will use other types of embryos and they'll upload them and they'll get, fair results. And they want to know kind of how the model's performing. Because we have all these different models. I know that Ujjwal was working on this last summer. We have a number of different models that we have available that we can use. So, Open DevoCell was one from 2 years ago. This is another one. We have the model, sort of the DevoLearn Classic from last year, and those are all kind of like, options for us to use. And so, sometimes in some embryos it might work really well, and sometimes in some embryos it might not work well. And we want to know kind of what the statistics are for it. And I don't know how, if you're like, if you're knowledgeable in machine learning, that's probably useful to you. If you don't know much about machine learning, then people are just going to use their eyes to look at it and say that looks good or that doesn't look good. So that's something that we can have, just as depending on who's using the interface, that might be useful or not. So yeah, thank you. That was a nice mock-up that you made here. I like it's a fairly clean mock-up. You have I would imagine you'd have like a directory here where you'd have images and you could just choose to analyze them, or you could do a bulk analysis or something. Yeah.
Actually, in fact, you could also drag and drop any image in that web app itself. So it's not just these example images. It actually is a working model in the background. So if you have such data in your computer, so you can just drag and drop some image in the box and you'll get the output. So it's not just a mock-up. So yeah, it's actually the working model in the background. So yeah, that's what I wanted to add.
Oh, good. That's great. I wanted to ask some more questions about your blog post here. So I was looking at these images yesterday, and they look very good. I like the way the algorithms being able to pick out things from the noise masked input. So like you have some noise over the top of it, and you have the images coming up at different intensities, and it's able to pick out it's able to segment some things that look like cells, and even like cell divisions if you watch it.
Yes.
So that's nice.
Yes.
That's good. Now, this the z-axis is like you're looking at the different stacks. So like they have a z coordinate in the data set. They have a z-axis, which is like going through the stack, and it's like the dorsal ventral axis. So this is just going down that axis and looking at how the images or how the values vary on that axis, correct?
Yes, exactly.
Okay, good. And then time is basically taking images and just running through them over time. So you're not running through a stack, you're running through time.
Yes.
Okay. So that's
and
interesting.
It's the same slice in the z-axis. Like here the z-axis doesn't vary, only the time axis varies.
Right.
So it's the same slice, but for a different point in time for each frame.
Right. And so yeah, you kind of see a different pattern of like how things are popping up. Like, like the z-axis, if you go through a stack with a microscope, you'll see like things kind of come into focus, come out of focus, and then, as you go through.
Yes.
And that's, you kind of see that here in the top image. In the bottom image, you see things kind of emerge in time, like their cells are dividing or popping into view. Yeah, so that's good.
Yeah, so in the image below, we also see some cells, they do pop in and out of existence, because like in the segmentation map, actually, like it might look like cells are popping in and out of existence, but actually what's happening is that they're actually moving away in the z-axis. So
right.
if they're far away from that plane, then the exposure is very low for that cell. So the segmentation map, it really doesn't pick up those cells which are moving away in the z-axis. If they're small enough, or if they're far enough in the z-axis from that plane, then the model just doesn't pick it up. So that's what is happening.
Yeah, and so it gets to a certain level of blurriness, and it just yeah. So that's good. But it's good that you have this noise mask, and you can actually cut through a lot of that noise to get the.
Actually, what's happening, like the noise mask, as you're talking about, like the data that we usually get from this, like that the data that we usually get, they actually contain noise. Like usually the data is not actually perfect. So I guess this, like if it's able to work with these noisy samples, then I believe it will also work with actual real world data. And this data is actually very close to real world data. So there's really nothing to worry.
Yeah, it's very good. Very good. All right, yeah, that's good. So that's all I had. Anyone have anything else they wanted to mention before we move on? Okay. All right, so now let me go to I'm going to talk about our submissions briefly. And then I want to try the entire screen share. Okay, you can see my screen now?
Yeah, we can see this.
Okay, good. All right, so this is our submissions document. So we update this periodically. There was a period where we were updating things every week, but we're not submitting every couple days like we were earlier this year. But anyways, let's go through this. So we have a number of things we want to kind of go over. A lot of things completed. The DevoLearn paper is going to be later, maybe in the fall after the end of GSoC. The Basilarian Non-Neuronal Cognition paper is still going. I'm working on it. I'm getting it to sort of a critical mass now. It's just been, kind of, you don't want to push these things too hard because, you have to do a lot of thinking and getting things in order. So this will be forthcoming. I don't know exactly when, but it's coming along pretty well. It's not going to be too much based on data, as it turns out, but that's okay. This particular paper, so we'll probably maybe have another derivative paper with more data, but I didn't want to make this dependent on a data analysis right now. So this is just something that's been solicited for us, and I'm trying to get the basic ideas out there. We have the boring billion in the Kindle book. So the boring billion is this idea of what embryos looked like a billion or more years ago. And we've talked about that in meetings. And so that's still outstanding. Maybe we can turn that into some sort of submission. The Kindle book is something that Krishna was talking about working on, but that's kind of frozen right now. The ANNs, BNN stuff, although that the particular paper, which is now on the archive, was rejected from A-Life, we still have it on archive, and we're still kind of pushing on that area of research. We have that. I think Akshay last week asked me for some material, so I sent him the paper and I sent him the talk at Net Neuro, so he's looking into that. We'll probably be working on that some more in the near future. The Mathematics of DevoWorm, that's this poster that we talked about several weeks ago. This is just a collection of, sort of mathematical tools that we use. And some of that was in the open room poster that I showed a couple weeks ago. But this is still ongoing. We want to make this into something that's like a short paper and not just a poster. So the test of Williamson symbiosis, that's where we take these ideas of Williamson and use genetic data to test certain hypotheses. And we'll talk about Williamson in a little bit, but we have this. This is something that can be worked on. We have this some more things on diatoms, which are Basilaria and maybe some other types of diatoms. We have these molecular level simulations. We have movement simulations, so you're looking at microscopy data and you're trying to discern the movement and maybe even the higher level movements. So movements are composed of different derivatives of position. So you have like motion, acceleration. Then you get up to higher level derivatives, which are jerkiness and other types of processes. And so these are like things that are very high frequency movements. And so that's what this is about. This game theory and developmental processes, this has been accepted at Dynamics Days Europe. This is something that I'm going to be kind of putting slides together on soon because the Dynamics Days is in late August. And we'll talk more about that when I put the slides together. This is one of the open papers that we have. So this is something that's open if people, when they see the presentation, want to start contributing to, we can talk about it. But I just wanted to mention it now so that people kind of have it in their minds and get prepared. Then we have this quantitative comparison of Archaean-shaped droplets. That's the idea that these Archaea bacteria, which are single-cell organisms that are not bacteria, they're their own group in the tree of life, that they form these different shaped phenotypes. They can be triangles, they can be different shapes, and then you can compare them to shaped droplets, or as was done in this Euler Paths for Life presentation, you can analyze them using a network approach. And so that's kind of what we're going to do for this. But this is something that needs maybe some initiative. I think we'll get there eventually. It's just we need, if someone's interested, we can talk about it. It might bump up the priority of it, and that goes for everything. If people see something maybe they're interested in, they can let me know, and we can sort of work on it as a higher priority item. So I talked about Steve McGrew 2 weeks ago, and he was what they call a polymath, and he did a lot of things in his life. And one of the things is he wrote this book that was unpublished, and it was called Eye of Nature. And I actually have a copy of the manuscript. I was reading through it in the last couple of weeks, and it's really interesting. It's got a lot of interesting ideas. It's about evolution and about sort of biology and there's this non-neuronal cognition component to it as well. So, these are things that fit well into the theme of our group. We're thinking of maybe publishing this as some sort of, self-published thing, or maybe publishing it in pieces. It's really hard to say what will happen, but I think we'll maybe closer to the fall, we'll start going through the chapters and looking at what's there. And finally, there's this differentiation tree of the brain, which that was mainly a note for me to bring that up in a meeting, and I wanted to bring it up when Dick was here, but I'll wait for him. I keep putting it off because I'll bring it up in a couple weeks. But this is basically some stuff I was doing with my other group on computational agents and how to develop them and evolve them. And so there's some insights here. A differentiation tree, of course, is where you look at cells as they differentiate, and they form these tree structures where, one group of cells differentiates into a tissue, and then that gives rise to other tissues, and so forth, and it builds this tree or this sort of hierarchy in time of tissues that form. So I'm going to talk about that in a couple weeks. So these are all different things that you can, if you're interested, let me know, and I can give you a deeper dive into the topic. We have links we have deliverable links. This hasn't really been updated very systematically, but I can give you the latest updates on that if you're interested. Oh, hello, Akshay. We were just talking about you. I was talking about the ANNs and BNN stuff.
Oh, I went through the paper. It is pretty interesting. Like, I didn't get anything to add. It pretty much covered whatever was required. So
yeah. Well, we'll be updating it, and that's a fast-moving area. It's something that I don't know what we'll be doing with it, but we have in an archive paper you can update it and then put up new versions, and then eventually if you get it published, then that's a publication. But then even then you can make new make new things from it in terms of research. So yeah. I mean, if you think there's things that maybe I mean, even if it's not now, it could be, oh yeah, Ujjwal, I'll share it with you as well. If there are things you may think of that are interesting concepts or interesting ideas, you can bring them up and we can see where that leads. I think there's a lot of interesting stuff going on at that interface. So I think, yeah, that's good. Yeah, I'm doing Neuromatch, and one of the groups I'm working with is doing they're comparing the visual stream, which is in the human brain, which goes from V1 to V5, and then there's some higher order centers. And they're looking at using an AlexNet and another, I think, a VGG version of a VGG network to look at, if you compare the levels, if you break the visual stream down into levels and you look at an AlexNet, say, at their different levels, what is a representation? So for V1, which is at the top of the visual stream, does that correspond with the top levels of an AlexNet, and so forth? And so there are different analyses you can use to look at the way those things compare, whether I think you're using a correlation coefficient, which is maybe not the best tool, but it's what we have now today to look at the similarities. And the similarities aren't very strong overall, but you do see patterns. So some of the levels are doing things more like the brain than others, and it's really interesting.
Yeah.
So yeah, I'll share that with Ujjwal. All right. So, any questions on any of that? Okay, I'll go. I'm going to talk about our task board, since we have a new person today. And this is kind of a longer version of what I showed you, but these are broken down to specific GitHub tasks. And they're not coding tasks necessarily. They're just things that issues that we have that we address from time to time. So we have, things that are finished for the year. Makes you feel accomplished to have a long finished list. But, these are things that we put on the board and they're finished. We also have these things that are off the radar. So there's a lot of stuff that's stuff that we were going to do, it didn't happen for whatever reason. This actually might be back on the radar here, because we have this talk. So see, things fall off the radar and they come back on. And then things fall off the radar and you get things that are held because you don't know how to implement them. So this is how it's organized. It's like we have things to do and we kind of propose them. And then maybe we move this into hold because it's, something we don't really know how to do, but it's still kind of interesting. Then we have action items, which are the things that are, more in process. And then in progress are things that we need to do to get it in progress. These are things that are in progress and then finished. So let me put this link. I'm not really sure if there's anything to update on this. Okay, Mainak, thanks for attending. Okay, so I think I did put that in there. All right. Oh, yeah, I had it in there. Okay, good. So that's the link to that. And this is the major task board. So again, people are welcome to propose things on this board, and we put it here, and then we move it across. We have a couple things, like if you're looking for projects, we have some things here, and it doesn't really tell you, too much about the project. It just gives you a taste. Like, for example, number 25, Axolotl embryo animations and segmentation. So this is something that is something we've been looking to do for a while, and there's this thing in the OpenWorm Foundation. They have a Docker container where they have all these different representative simulations of C. elegans. And so our contribution would be an embryo. And we've been really kind of trying to figure out how to do this. And it hasn't really happened because it's very hard to do. It's very hard to get the right kind of thing going. So, it's something that if you have an interest, we can talk more about it. And we can do something. We also have other things that are kind of off the radar. We do a lot of stuff with imaging or with microscopy imaging. We have an interest in neural organoids from time to time. We talk about different papers, and we've been trying to do this thing where we're using secondary data to analyze. So neural organoids are these cultured groups of cells that form these neural-like structures. So there are a bunch of like neural stem cells, and you culture them in the right conditions in a three-dimensional cell culture, and you can get them to form shapes. And the shapes and patterns they form are not really like a regular organism, but they're organoids. And so people have actually used these to, study neural function. They've used them to study development. So there's a lot of interesting data in there, but we haven't really gotten that off the ground. And then there's this stuff that we're doing on Basilaria and the Basilarian non-neuronal cognition, which is where we're positing that Basilaria, or these colonial diatoms, and it's just a bunch of cells that are moving together, that they have this sort of cognitive aspect to their movement. That's what they call non-neuronal, meaning that there's no brain. It's just some sort of model of movement, some sort of model of like information processing. And I'll cover that in a couple weeks, but suffice it to say that this is something that it's not an easy thing to kind of convince people of. So we need to have some solid computational models. We need to have some data analysis. So it's that's coming together slowly but surely. And so that's a couple issues around that here. So again, that's what we have for people if they're interested in contributing. And now I think I'll go to the papers folder. And in the papers folder, we have a lot of interesting things to talk about today. So we didn't have any more comments. Well, where we have comments in a little bit, you can put them in the chat. So today we're going to talk about a couple of things, a couple of themes. First is we're going to revisit this idea of metamorphosis. And I found a couple of interesting papers this week on this. This is a topic we've talked about in the past. How does a caterpillar turn into a butterfly? So metamorphosis is where your organism is like in one state, and then there's this period where it goes into a cocoon, or it goes into some sort of hibernation, and then it comes out in a totally different state. So, for example, the caterpillar turning into a butterfly. The caterpillar is this long worm-like creature, and it's crawling around, and then it forms a cocoon at some point in its life, and then it turns into this butterfly as it's an organism with wings, as it's in the cocoon, and then it hatches and it goes off. And so we've talked about how this happens developmentally. You have this developmental program where you have this caterpillar formed, and then it goes back into sort of this developmental stage where some of the cells, when it gets into the cocoon, they decellularize, de-differentiate, and then they differentiate in different directions. And that's how you go from this sort of, worm-like creature to something with wings. But they talk, these are two really kind of accessible articles on this. So they say, what does this radical transformation entail? How does a caterpillar rearrange itself into a butterfly? What happens inside a chrysalis or cocoon? First, the caterpillar digests itself. I'm sorry about this. I had printed it from the web and there's text over it, but releasing enzymes to dissolve all its tissues. If you were to cut open a cocoon at the right time, caterpillar soup would ooze out. So it would be like this, just the soup of whatever's in the, that's going to become the new cells and tissue. But contents of the pupa are not an amorphous mess. Certain highly organized groups of cells aid in the digestive process. Before hatching, when a caterpillar is still developing inside the egg, it grows something called an imaginal disc. So for each of its body parts, so this is something we see in Drosophila, which is the fruit fly model. It will need as a mature butterfly or moth. So it has these like developmental sort of things that happen in the egg before it becomes a caterpillar even, that are going to be important for the butterfly phenotype. So they have these little discs that define different parts of the body. In some species, these imaginal discs remain dormant throughout the caterpillar's life. In other species, the discs begin to take the shape of an adult body part, even before the caterpillar forms a chrysalis or cocoon. Some caterpillars walk around with tiny rudimentary wings tucked inside their bodies, though you would never know by looking at them. So they have this imaginal disc, which is so in the fruit fly, the imaginal disc might begin with only 50 cells and increase to more than 50,000 cells by the end of metamorphosis. Depending on the species, certain caterpillar muscles and sections of the nervous system are largely preserved in the adult butterfly. One study even suggests that moths remember what they learned in later stages of their lives as caterpillars. And I think we've talked about this in previous meetings, but the flatworm, which is a model for regeneration, it's a different type of worm than C. elegans, but the flatworm actually has this incredible regenerative capacity where you can take a single cell and you can create a whole new organism. And these cells are basically totipotent, meaning that a single cell can reproduce the entire phenotype. And what's interesting about flatworms is that if you take a single cell and you regenerate a phenotype, that new phenotype will actually sort of reproduce the memories of the worm in which it was taken from. So there's this flatworm, you take a cell out, you make a new worm, and then that worm retains the memories of the old worm. So it's interesting that mechanism sort of may exist in both of these systems. So getting a look at this metamorphosis as it happens is difficult. Disturbing a caterpillar inside its cocoon or chrysalis risks botching the transformation. So they actually have some incredible photos of this silk moth that failed to spin a cocoon. So this is the link to the incredible photos. And so we can see here that, like, these are eggs. This is the caterpillar as it's crawling around. After about 5 days, the hatchlings begin to shed their first skins. Then the second instar caterpillars, which is just the developmental phase, has grown quite a bit. Then they kind of do this molting where they go through different stages of development. And then they finally get to these different versions of caterpillar. So they kind of go through this. Now they're starting to get into this. The caterpillar doesn't make a very tight leaf raft before beginning its cocoon. A lot of it is open to the air. And then this is the completed cocoon. So once they get to a certain stage in development, then they form this cocoon, and it tightens up over time, and it forms this thing here. And this is just a protective shell. It's like paper mache, so it's very soft. It's very brittle. It's not soft, but it's. I guess if you could squeeze it, as a human, you could squeeze it apart, but. And then so this was previously the caterpillar of a Tusa moth, which is a certain type of transformation here. The caterpillar has already shortened up, becoming a prepupa. Inside the caterpillar shell, it was developing the proto-versions of its moth organs, antennae, wings, and legs. The prepupa is incapable of walking and can only wiggle in midsection like a hula dancer. So these caterpillars that would walk around and now have lost their legs and they can't walk at this stage. They're just kind of wriggling around. It begins by shrugging its shoulders in this area, back and up, splitting the caterpillar skin. Unfortunately, these shots didn't turn out, so it was a little bit blurry in this one. This is where it's sort of mid-pupa. It's like coming out. It's moving around a lot when it's in this stage. And the antennae and wings are actually preformed, but they're not complete. They are separate. And then this is it's very hard to image an in-between moment. So you get they get this transformation, but I don't think anyone's been able to really capture sort of very precise images of this process. It's just that they have this sort of hardware on board, and then it eventually becomes these things. But it's very hard to see inside the pupa to see this like a time lapse. We can't really acquire that here. And so then we can see the shapes of the legs, antennae, and wings distinctly here. The entire thing looks like it's been carved out of delicate green jade, but this is the sort of the skin here. So it's still not looking like a butterfly, but we're getting there. Within about 5 minutes of eating totally free of the caterpillar skin, the pupa is closed in and the free-floating antennae and wings are attached firmly to the sides. The pupa will darken over a matter of hours, and then you get this emergence of a moth, in this case, which has wings, and it has this basic phenotype from the caterpillar, but it has wings and it has different parts that were already kind of there as precursor organs. And they were developmental organs that just kind of differentiated through this process. So that's this article. And then there's another article where another Scientific American article where they kind of talk about, what people are doing to find the origins. They do bring up Donald Williamson, and people kind of ridicule some of the ideas he had. He had this idea that butterfly metamorphosis resulted from an ancient and accidental mating between two different species. And the reason that he says that is because you can see it looks like two different developmental programs. But people have ridiculed that idea because there's no real evidence for it, and that it's maybe kind of a little bit, doesn't really fit the data that we have. So how do we study this? Well, we could use like molecular data, which is something we've proposed doing. But it's maybe more important to understand the actual process, which is why I showed you all those pictures to give you an idea of what this process looks like. And so, maybe it isn't that you have two different developmental programs. Maybe it's that you have a single developmental program that has many parts. So we don't really know how this works mechanistically very well, but this kind of goes through the history of like how people have thought about metamorphosis, and this goes back to the 17th century. So people have proposed a lot of ideas about this, why you get this transformation. We know that there are imaginal discs involved, and we know that there's certain genes that get expressed. So we know that there's certain things that are going on, but we still don't really have a good time-lapse imaging of it, and we don't know much about the genetics. So so this is just kind of goes through a lot of the state-of-the-art. And we do have evidence, but not really very conclusive evidence. So that's, I'm going to send this group of papers out for people to go through if you want to go through it more closely. Okay, so then the second thing I want to talk about is, last week we talked about what is environment. And this came from a question that I had that Dick and I were talking about after the meeting one day. And, we talk about environment as a factor in development. So environment is just the things outside of the egg or the pupa or whatever, and the influence. So you can have, last week we talked about light and the quality of light. And, of course, temperature can also play a role in influencing development. So you have these stimuli outside the egg. It could be changes in temperature or changes in light or specific types of light. So if I bring up the questions that we had last time I talked about this, we had a question: what is environment? What is its relevance? And how do you define it? And so we came up with these two categories. One is that environment is permissive. This means that there's this general information in the environment, so changes over time, stresses, intensities, these gradients of temperature are transduced into the biological system. So a change in temperature is an input into the biological system. It might trigger some gene to be expressed or some response, some stress response. The second is that environment is instructive, and that's that specific information such as ratios, patterns, and codes are then transduced into the biological system. So they could be environmental patterns, like if there's a drought, or if there's this ratio of light to dark, or something like daylight to nighttime. So if as your seasons change, daylight might change the amount of daylight that an organism gets. And so that might have some effect on the biological system. So like with different types of circadian rhythms and that. So these are two different classes of problem. And so today I'm going to talk about this citable article from Nature Education. It's called Physiological Optima and Critical Limits. So this is talks a little bit about like temperature gradients and like sort of how we think about like optimal conditions and like what are the limits of those conditions that trigger changes. So organismal distribution limits and responses to climate change depending on how physiological performance varies as the environment shifts between optimal and extreme conditions. So, there are periods of Earth history like in the Pleistocene, which was, several thousand to a million years ago, where you had these shifts between like glaciations and warm periods, and that affected climate all over the world, where you had these shifts in very rapid shifts over maybe about 200 years, where it went from like very dry to very wet, or from very cold to very warm, depending on what latitude you're at. And certainly today we have places where it's getting much warmer, and that organisms aren't really adapted to that. So these are things to be to think about. So distribution limits of organisms are dependent on biotic and abiotic environmental factors. Organisms maximize their fitness at an optimal environmental range. So they kind of, they respond to the changes in environment or the variation of environment by trying to maximize their fitness. But they try to find this optimal environmental range. But they can only survive short periods in environmental conditions that exceed a threshold in their critical tolerance limits. So here they have a picture of penguins who live in Antarctica. And, imagine if they were transported to the Amazon rainforest where it's very hot and wet. And their physiological tolerance in that case would be very limited, because they're not, their fitness is sort of optimized to this sort of, Arctic environment. And so this is the where they've evolved their abilities to tolerate temperature and things like that. If you move it to another part of the world, they may not do very well. So this is what they're talking about. Breadth of optimal environments and the range of environments between critical limits vary depending on physiological adaptation and energetics. So, they might have to conserve their energy if they go to a new environment, if there's a change in the environment. Or they might have to adapt, if they can, new ways of dealing with that. Critical limits may serve to define species distributions, community structure, and how communities respond to environmental changes. And those are ecological terms that basically relate to the ecological niche that they inhabit in the structure of their populations. So variation in fitness between environmental factors that define an organism's critical limits can be diagrammed as what they call a performance curve, the shape of which can be interpreted in the context of physiological mechanisms at the organismal and cellular level. So there's this ability, so they show a thermal performance curve here with temperature. So you have this lower critical limit. This is performance, which could be like behavior, could be like reproduction, it could be anything. And then this temperature gradient. So you have this lower critical limit and this upper critical limit. And then you have this performance optima. So when the temperature is very low, the performance is limited. If it's like a human and it's like, 10 degrees below zero, humans have to be in a warmer environment. They have to wear clothes. Performance is limited. Similarly, if the temperature is like 125 Fahrenheit, certainly at 125 Celsius, that would be beyond the upper critical limit. But the point is that when it's very, very hot, people can't perform optimally unless they're in like a building with air conditioning, or there are ways to get around this. But generally speaking, there's a performance optima that we seek out, that we do our best work in, as it were. So this is what they mean by the influence of environment. And so the reason I bring this up is to show these performance optimum curves, to show that you have this thermal, like in the case of temperature, you have this thermal gradient where there's an optimum point, and then there are these suboptimal points, and then these critical limits that define sort of the edges of the survivability of an individual organism. And in this case, they're talking about fitness. So in terms of if you're below or above the critical limits, you have no reproductive fitness essentially. In terms of people living their lives, they might die or they might get injured or something. In development, when you have eggs or you have embryos in a womb, they're affected by this as well. And so they experience stresses during development, and these temperatures translate into extreme stresses or some sorts of stresses that are not optimal for survivability. So this has an effect on that as well. And so like in Drosophila, for example, temperature variations, fluctuation in temperature is very critical to the survivability of the eggs. And so when the eggs are exposed to really high temperatures, they can develop a lot of phenotypic mutations that are deleterious, that are bad for them. If they're at the optimal temperature, however, they seem to be, they develop without any incident. So this is why temperature is important. So again, this is this temperature input coming into the system and changing things for the organism. So that's spelling out that aspect of environment. Now I'm going to go through some maybe two more papers, and I think if you have things in the chat, if you want to put them in the chat, we can talk about them. Let's see, I have this. This is from a couple weeks ago. We talked about cephalopods or octopus, and so we talked about how octopus have this really unique brain and how it develops. So octopus brains are a lot different from human brains, but they have a really fascinating developmental trajectory. And so in this paper, they're talking about how cephalopods actually can serve as a model of cognition. And so we've talked about non-neuronal cognition, which is where you have this model of a sort of a brain where there is no brain, things that process information like a brain. In this case, this is a different model system for cognition. We usually think of the human brain as a system for cognition, but cephalopods also are very intelligent, or let's say express these benchmarks for intelligence, but the brains are totally different. And aside from being connected into a network, they are they're very different, and they don't have the same aspects. So this is something you think about when you think about neural nets. Neural nets are generalized sort of human brains. They're generalized brain, mammalian brains. But there are other types of brains in the world, and so this is one of them. So traditional approaches in comparative cognition have a long history of focusing on a narrow range of vertebrate species. So in recent years, this range of models has expanded. So for example, invertebrates like C. elegans, you have Drosophila, which is the fruit fly. You have a lot of these where we actually know what the connectome looks like. So those are, but we don't really talk about those as being cognitive, although we do know that they exhibit behaviors. We also have other types of organisms that are, have like pretty decent sized connectomes that we really don't characterize as cognitive, and yet there's no reason why we shouldn't. So in cephalopods offer a nice balance because they do exhibit these kind of cognitive traits, but they also have a nice connectome or brain that we can look at. And so in this review, we contend that cephalopods are suitable ambassadors for rethinking cognition. Cephalopods have large, complex brains, exhibit sophisticated behaviors, and increasingly people think that they really have this complex cognitive suite of behaviors that are maybe more like what we might see in humans. So that means that maybe they do some, metacognition or things like that that are, that we think of more in terms of human brains. And so these are brains that evolved independently from vertebrates, so they look nothing like human brains, but they do have these same properties. And so I don't know if there are any good pictures in this paper, but So this is a species of octopus here. Another species of octopus. So this is their social organization, cephalopods. So there's a solitary day octopus at the top. So these are solitary organisms. The middle frame is the broad-club cuttlefish, which is competing for mates. And at the bottom we have this Caribbean reef squid, which is a social species. So there's a wide range of like social behaviors and solitary behaviors. And they have this idea, one of the ideas about why human brains are so big and have so much cognitive capacity is this idea of the social intelligence hypothesis, which means that if you live in these social settings where you have a lot of individuals around you, you have a bigger brain because you have to remember the relationships. Now that hypothesis is somewhat, it's not absolute, and we can find exceptions to it. But we know that social pressures can influence cognitive development and performance within species. So there's this selection that goes on for larger brains. And so you see this in octopus, but even the solitary species seem to have pretty decent sized brains. So, this doesn't explain all the variation, but there is this link that they talk about here between social group size and cognitive performance. And so I don't know if I don't have any images in here, but they just kind of bring this up. So I thought I'd bring this up. They don't have a lot, like in the last paper I showed, they had a lot of images of the brain itself and the anatomy. In this paper, they just talk about some of these aspects, sort of advocating for cephalopods being a good model for cognition. And so, let's see. This is another paper that, this one talks about the integrative neuromechanics of crawling in D. melanogaster larvae. So these are the fruit flies that we talked about, the Drosophila model species. And so this is a model species that differ from C. elegans in a number of ways. They have more cells in their body. They have, bigger, more complex brains, relatively speaking, and they have these imaginal discs that are sort of the basis for their development. And so they have the cellularization phase, and then they have these imaginal discs that emerge. And so the abstract of this paper is locomotion in an organism as a consequence of the coupled interaction between brain, body, and environment, motivated by qualitative observations and quantitative perturbations of crawling larvae, which means they looked under a microscope and they did things with different types of perturbations at different dosages maybe. We construct a minimal integrative mathematical model for its locomotion. Our model couples the excitation inhibition circuits in the nervous system to force production in the muscles and body movement in a frictional environment. So they're forcing this sort of movement through stimulating the muscles. So they're getting body movement, and I guess they're modifying the environment. Then it's linking neural dynamics to body mechanics. So they're actually looking at the mechanics of the behavior, and they're actually looking at this in development in larvae to see how these things work and how they change in development. Our results explain the basic observed phenomenology of crawling with and without proprioception. So proprioception is where you're sensing forces and sensing the external world of physical sort of forces that act upon the body. When we walk, on different surfaces, we can tell that we're walking on different types of surfaces. That's proprioception. And so this is something they can modify. This is an environmental input they can modify. And elucidating the stabilizing role that proprioception plays in producing a robust crawling phenotype. So more generally, our approach allows us to make testable predictions on the effects of changing body-environment interactions on crawling, and serves as a step in the development of hierarchical models between cellular processes and behavior. So they have this, so they want to come up with this sort of complete theory of locomotory behavior. They need to characterize movement. They need to characterize this in the larvae. So they have this GFP imaging of larvae So they're looking at the forward crawling body segment and gut movements. So these are gut movements, or what's going on in sort of the digestive tract, and the body segments are different parts of the phenotype that allow it to crawl. And so, that's what they're looking at here. They're putting aside the mathematical details for now, but this is a good figure that shows kind of what's going on here with their model. So this is the phenotype of the larva. So this is not the embryo, but this is the larva, which is hatched. And you can see that they have these different segments from the head to the tail. So this is the head and this is the tail. And there are 10 segments here. And so each of these segments, and I think we've talked about Hox genes, where there's like a certain Hox gene that's assigned to a certain segment, and that they give rise to, you give rise to these 10 segments, which are based on the same sort of template, but then they can be varied through evolution or through whatever Hox gene is being expressed. So there's some variation between these segments, although they all come from sort of the same place developmentally. Then this is their model here in B of one segment. So this is a model of a segment where there's a neural controller, there's excitatory input, then you have this. So this is the anterior segment, which is the segment to the front. So if we take 5, for example, 4 is the anterior segment, and then the posterior segment would be 5, and then further posterior would be 6. So the anterior segment has this neural controller on it, represented by a neural controller. Then the posterior segment, which is 5, has a number of things in it. It has the stretch receptors, soft body mechanics, muscular forcing, and of course friction between the segment and the environment. So this friction is where the proprioception comes in. The friction, depending on the strength of the friction forces, they get transduced into the body, and it serves as a signal, feedback to the neural controller, but also to some of these mechanics. And they sort of work together to determine the movement of the organism. What we've done, there's been a lot of work done on this in C. elegans, in the OpenWorm Foundation. We have the cybernetic project, and they kind of think about this model of like body mechanics in terms of C. elegans and a lot of the friction forces that C. elegans can experience. And then C is this larva body is modeled as a linear chain of masses connected by damped linear springs. So this is typical of a neuromechanical model where you have this set of damped springs between the segments. That means that there's a loose coupling, and there's this sort of these physics between the segments. So, it's kind of like where you have a connection, but it's flexible. So it's kind of like an accordion almost, where it doesn't break, but it doesn't also it isn't rigid. And this is important in mechanical systems. And then you have this sort of this neural controller in each segment. And so we saw before that this anterior segment inputs to the posterior segment, but each segment in turn has a neural controller. And so you have this integration that goes on across the body. And then you have the environment, which is this solid friction, these friction forces. And then finally D is this is the segment-to-segment propagation of neural activity. And they're trying to model this as it happens through neural and proprioceptive couplings. So the neural couplings are this WEN here, so these E's, these circles with the E's in them, there's a connection here. WEP and WIP, these are the proprioceptive couplings. So this is coming from contraction detection so you have motion, you have this contraction detector, you have this muscle drive. The muscle drive is outputting from the neural system, so from E, that gives muscles like some signals to move. You have contraction, which is the proprioceptors detecting whether the muscle is moving against the surface, and then that's feeding back into the neurons and into this I, which is an inhibitory neuron. And the excitatory and inhibitory neurons in this segment are working together. There's an inhibition going on from the feedback. There's excitation, feed forwarding from this neural element to the muscle and to the inhibitory neuron. And then there's this feed-forward signal from one segment's excitatory neuron to the other, and then there's this contraction detector which goes to the anterior neuron. So this is basically their model for modeling movement behavior and the what they call the neuromechanics of it. So that's their basic model, and they walk through the math. So there's a lot of modeling that's done here. They use Wilson-Cohen equations, which is basically a neural model which accounts for excitatory and inhibitory populations of neurons in a segment. They use this over phase oscillators. And when you build a mathematical model, you have to make a lot of choices, like you do in machine learning. And so you make these choices and you rationalize why you make them. So this isn't the right answer necessarily, but they make these choices based on the literature, based on what they're trying to achieve. So, and then body mechanics. So they talk about passive tissues, which are these soft tissues, and I'm hoping that Susan can give a talk on this soon. She mentioned this a couple weeks ago. I don't know, but soft mechanics and soft materials are really interesting biological implications for those. So so if you're interested in this paper, I can put it up in the Slack, and they give a lot of results here of how they run their model on different parameter values, and this shows you distance of crawling, muscular activity, and neural activity, so they can check all these things. And so they even have links to videos in here if you're interested in seeing what these looks like in terms of displacements and in terms of the muscular force. So I think that's it for today. I hope you've enjoyed this. It looks like everyone had to leave. Okay, so Ujjwal had to go, Sanjay had to go, Akshay had to go. Okay, so let's see. Yeah, so Sanjay said, being my first DevoWorm meeting, I got to learn about interesting research works of the community. Hoping to work on something to present one day. Have a great week. So have a great week, Sanjay and Ujjwal and Akshay and Mainak. And everyone have a good week. We'll be on Slack and on GitHub, and hope to see you around.